Hi, this is Shelley Kraft, and welcome to SNNLive. We're here in San Francisco at LHA. I have Dr. Claire Kruger of Spherix Inc., SPEX on NASDAQ, standing right next to me. How are you?
Very good, thank you.
It's good to have you. I'd like you to tell the audience what Spherix does.
We're a biotech firm that's in drug development. Right now we're looking at drug candidates for use in treatment of metabolic disease, particularly for lowering of dyslipidemias like high triglycerides and high cholesterol. We also have a drug candidate that's gone through phase 3 clinical trial for treatment of type 2 diabetes.
So, finished phase 3. So
Yes.
That's ready to come on the market pretty soon, I guess.
Well, the drug that we have that completed the phase 3 was for treatment of type 2 diabetes, and as you may know, FDA's regulatory climate changed dramatically in the last several years due to cardiovascular events for drugs in that category. That makes the safety burden very, very high for many of the drugs in that category, and really makes the development very expensive and much longer than typically it was several years ago. That's what induced us to look for other efficacies for a drug candidate and also for other drugs that we have in our current pipeline to look for what we believe is a much more commercially viable opportunity for us, and that's in the treatment of high cholesterol, high triglycerides.
So tell me, how does your product work for diabetes 2?
We believe that it acts as a sugar blocker, basically. It's the same mechanism by which it also impacts, likely that tagatose, the lead candidate, would impact metabolic syndrome or high fat, high triglycerides, high cholesterol. Because tagatose itself is a sugar, it's handled by the body very similarly to other sugars, and yet there are some important metabolic differences in the speed with which your body can metabolize it. And blocking these metabolic pathways produces transient accumulations of metabolic intermediates that help your body to actually store glycogen longer, release it more slowly, and that actually cause your body to more slowly convert your sugar or glycogen stores to fat.
So it looks like you're attacking some pretty large-sized markets.
Yes, we are. We do have in our pipeline and used in conjunction with tagatose SPEX 106, which is our other drug candidate for use in the hypertriglyceridemia market. It actually works very interestingly through a mechanism that's very different than tagatose, where tagatose might act to actually inhibit the formation of fats. SPX 106, we believe, works by increasing the catabolism, the breakdown of fats. And so both together function better than either alone. And as you know, the markets for high triglycerides, high cholesterol, dyslipidemia are very large and underserved right now. Many of the drugs that are currently in therapy have other issues with them that don't make them ideal for treatment of patients with those diseases.
What's your background?
I'm a toxicologist by training and did my postdoctoral work in biochemistry and come out of a consulting field where I worked with drug and food companies for 25 years, helping them get their products through regulatory approvals.
So what does 2012 look like for you guys?
We are just finished up the work that we believe is going to be needed to file an IND for our new drug SPX 106T, which is the combination SPX 106 and tagatose for treatment of hypertriglyceridemia. We hope to get that IND in and begin our clinical trials in 2012 to start producing some very exciting information about this new drug candidate.
That sounds good to me. What is your website?
www.spherix.com.
And it's probably one of those full of information websites you can, you know, like if someone wants to find that information about any of your drugs, you've got white papers there, you've got the whole story, right?
Yes, we have a very informative website and we encourage everyone to visit it.
Ladies and gentlemen, Dr. Claire Kruger from Spherix Inc., SPEX on NASDAQ. Claire, I want to thank you for coming on to SNN Live.
Oh, you're so welcome. I appreciate it.
Thank you.